So, well, I'll take you through the cartoon first. This is for recruiting to graduate school. Department Recruitment Statistics. Hard to believe, not according to the official department brochure. They say the average PhD takes 5 years. Maybe they need 50. Yet nobody you know has taken less than 7. They say the number of incoming students each year is 25. That's it. But only 4 people get hooded at commencement each year. They say the student to faculty ratio is 4-to-1. You go, no you go. So why does your advisor double book your meetings? They say the department brings in hundreds of millions of research dollars a year. Maybe if I get the cheap ramen, but you wouldn't know it from your stipend check. So this is academic research. Lab is run by what they call a principal investigator, primary investigator, number of different terms. But the research is generally conducted by the graduate students in his lab or her lab. Sorry, mine was a male, so I was thinking of the his. Okay, we're going to do one of these things where I'm not going to write a lot. So I'm not going to do too much on the overhead. Now you may want to follow along in the book because I'm going to follow the order of the book. When I put this class together, I was trying to come up with a way to tie all the stuff together in a way that had practical applications in the real world. And so I decided to do a cancer chapter because cancer, all the stuff we've talked about, or most of the stuff we've talked about, comes up when you get into the bits and pieces of cancer without necessitating going into really, really deep bits and pieces. So what is cancer? Because people go, if we can do this, why can't we cure cancer? Well, it turns out that you can define cancer, but it's not one thing. Is that when you have cancer, you have a population of cells that are just in the business of reproducing. They're just in the business of making more cells. And that business eventually drains your resources to the point where you can't survive. If cancer was in itself an organism, it would be a terrible organism because it has no way of reproducing beyond the host. And it tends to kill the host. There is such a thing as a contagious cancer. There's some cancers produced by viruses we'll talk about. There's a weird cancer that Tasmanian devils transfer from snout to snout when they nip on each other, where the cancer cells move into another individual and can then produce a cancer there, but that's pretty bizarre. That's pretty unusual. A tumor tends to be a term where you've got a bunch of cells dividing, but they're localized. They're in one place. When some of those cells start to migrate elsewhere and form new tumors elsewhere, that is metastasis or metastasis, and you have what's considered a malignant cancer. So where does this come from? It's a problem of mitosis. It's a problem of cell division. So it's almost impossible to have cancer that derives from a cell population where the cells don't divide. In your body, for instance, your nervous system is set in place pretty early on in your embryonic development, and very few of those cells ever will make more cells. They get bigger, but they extend processes, but they don't divide. So a brain tumor is not a tumor that started as nerve cells. It started as some of the support cells around the brain, which are more like connective tissue cells. When you get into the central nervous system, the tissues get a little weird in terms of the categories. But typically, you don't get it starting in muscle cells because your muscle cells don't form new, they get bigger, but they don't tend to form new muscle cells. So a disease that's predicated on mitosis out of control is going to happen in cells that go through mitosis normally. Now, in a lot of your organs, you have little pockets of cells that are just kind of in reserve that can produce replacement cells if you have damage. And in some places, you're always producing replacement cells. In your skin, since you're always losing skin cells, along the tubes of your digestive and respiratory system, since those cells are always dying and falling off, you've got a whole population in the underlayers that are making new cells. And so your most common cancers form in tissues where the cells are normally reproducing a decent amount, and something's gone wrong. In some cases, there are issues with differentiation. You make new cells, the cells are supposed to do their jobs, right? Interphase, they do their jobs. They don't do that. They don't form the cells they're supposed to form. They just kick through interphase and do another round of mitosis. And these cells are typically pretty damaged, but they're damaged in a whole bunch of different ways. In some cases, the spindles don't work properly, right? Going back to the mitosis we talked about not very long ago. In a lot of cases, there are mutations. You actually have a decent array of genes whose proteins are in the business of keeping your cells doing what they're supposed to do, making sure that you don't make copies of cells that are fairly damaged, making sure that mitosis doesn't get out of control. Some of the first cancer genes were actually cancer resistance genes, is that the proteins, when produced properly, keep cells from becoming cancerous, but when you get a mutation and the proteins don't work again, then you wind up with cancerous cells that are not under control. If you look at early cancers, I've got numbers here. It is very common if you take a population of early cancer cells, so this is something where it's just getting started, and you compare it to other cells from the same population that seem to be doing normal stuff. There was a paper put out, when I wrote this chapter, I read lots and lots of papers, that found between 1,000 and 2,000 point mutations compared to what the normal cell was supposed to have. And there were all sorts. There were substitutions, there were insertions, there were deletions. So what happens in a lot of cancers, and the reason why cancers tend to be associated with older, is that sometimes these mutations have to accumulate. It isn't one mutation in one gene. It may be one final mutation in a gene, like the straw that broke the camel's back. But it very rarely is a single cancerous mutation. Sometimes it is, but usually it's a collection. What produces the mutations? Mutagens, right? Chemicals that can produce, we talked about this in the history of genetics, is that when they discovered mutations and went, oh, how do we get these to happen? They found there's a lot of chemicals that do that. They found radiation does that. All of those can be contributory to cancer. It is true that a lot of cancer treatments actually, some of the side effects is they can cause cancer. But if you're heading toward dying from cancer, most people are willing to take that risk from radiation treatment or from an awful lot of the chemotherapies that are available. Is that you're going after cells that are dividing, you're also going to screw up a lot of normal cells that are dividing, which is why you get a lot of the side effects you do. We'll get to that eventually too. There's a new area now looking at the possibility of our native bacteria sometimes producing mutagens that can eventually produce cancers. Free radicals, good old oxygen, singlet oxygen atoms produce oxidative damage that can affect DNA, that can produce mutations. There's some viruses that can integrate into the host nucleus and mess with the DNA there. Cervical cancer is a viral produced cancer. There's a cancer that was an early clue that something was going on with HIV, human immunodeficiency virus, because there's a virus that can form lesions on your feet that is virus associated, that shows up just in people that are very immunocompromised and have an Italian background, if I remember properly. And, and there are hormonally induced cancers. Breast cancer very typically is hormonally induced, and a lot of the treatments that they put people that have breast cancer, they put them on tamoxifen. Tamoxifen is a hormone resistor, try to keep the hormones from getting at the cells, and then there are what they call hormone mimics, usually steroids, so entry into the cells fairly easy, and it's similar enough to the real hormones that they can get in there and mess stuff up. From the little numbers I've got here, the estimate is that maybe a fifth of cancers are connected to alleles that you were born with, that you inherited, right? Breast cancer, very famously connected to an inherited allele. But most cancers are derived, is that about 80% are alleles that accumulate through your lifetime. You didn't wind up in your kit to start with. About 2% of cancers are diagnosed before you're 40. Remember I said it tends to be an accumulation of issues. And by the time you hit the age of 80, about 50% of all diagnosed cancers are in people 80 and younger, which means 50% 80 and older. But then prostate cancer, this is not an exact number, but it's a scary enough number that I will pass it on. The odds of getting diagnosed with prostate cancer come very close to matching your age. So if you're 80, you probably have or have had an 80% chance of being diagnosed with prostate cancer. So as you get older, you get to be 100, you really dodged a bullet if you never got diagnosed with prostate cancer. The advantage to prostate cancer, if you can have an advantage to cancer, is it's very, very slow acting. The typical one, there's a nasty one, definitely have that dealt with. But your typical prostate cancer is very slow, doesn't tend to metastasize, and in people that are 80 and older, very often the discussion is, so if we leave this alone, it probably won't get bad for 15 or 20 years, and you're 83, there's a really good chance something else is gonna kill you before you hit that point. So, you know, we can go in, we can cut out your prostate, it can mess up your urination issues, it can make you incontinent, it definitely can affect your sexual performance if you're still doing that kind of stuff. Do you still want to have the surgery, given that this is unlikely to be the thing that's going to kill you? And we can keep track of it, you know, follow it anyway. So typically, older men with that form of prostate cancer kind of pass on treatment, which sometimes can be tough to convince their relatives, like, oh, granddad's gone nuts in the head. He's got cancer and they're not treating it. Well, granddad may have some good reasons for that. These cells are damaged. They tend to get more damaged as they develop through cancer. They produce more mutations. Those mutations can be chromosome duplications. It can be chromosome set duplications. The cells are fairly limited in what they're doing, so they can survive a lot of mutations that would kill an early embryo. They look a lot like early embryo cells, but they don't differentiate. So they're not dipping into as big a bag of genetic tricks as an embryo does, but they need to survive to be a successful cancer. So what you're going to find in later cancers is a lot of the same genes have been activated. There's a lot of epigenetic issues in cancer. Cancers activate and deactivate certain genes, and in order to be successful long term, you find a really advanced cancer very often has the same array of activated alleles, because those are the ones that allowed it to get to that point, whether it started as a lung cancer, started as a breast cancer, started as a bone cancer. Advanced cancers are very difficult to tell what they started as. You have to really dig into the genetics, because a lot of the basic genetics are very, very similar, because they're evolving basically. Other things associated with onset of cancer: histones, the DNA wrapping proteins, and how the DNA wraps on it. There are cancer starts that are associated not with changes in the DNA itself, but changes in how the DNA is or isn't wrapped up. They're what they call insulated neighborhoods, where the wrapping is fairly tight in a whole zone. if those unravel, you get what's called destabilization of insulated neighborhoods, and that can activate genes that normally would be left alone because they're wrapped up pretty tight where they are, and that alone can lead to cancer. Remember I said there isn't, this is why you can't go after one thing and eliminate all cancers, is that most of the stuff that you can go after that's one thing, the cancers are pretty well advanced and they're pretty well on their way to killing people, so it's much trickier to do. And again, it still doesn't work on everybody. There are transcription issues for some cancers. There are promoter changes in some issues. There are enhancer changes in some issues. Sometimes it's called hijacking of a promoter or an enhancer in order to activate a gene that shouldn't be activated, or deactivating it. There could be issues in the splicing process in messenger RNA. So everything we've talked about really can be connected to initiation of cancerous changes. I'm sure at some point the little bitty RNAs that are active in our cells that we're just starting to learn exactly what they do, I'm sure there's some ways they go bad where cancer is the ultimate product at the end. There is a type of change molecules I've run into. This was a really odd paper, but they were suggesting that there may be what they call super enhancers at play. And epigenetic erasers that pull the methyl groups off of your genes in order to get them to express when they didn't. Remember epigenetic changes. Lots and lots and lots of stuff. I spent a long time digging through the literature and then trying to take the technical stuff and digest it down into technical stuff for beginning biology majors, so stuff I expect you to understand, but tried to keep it from being so technical that that somebody couldn't come in and say, go through our book chapter and have some idea of how this all works. Okay, I've got, okay, well, we'll do this and then that'll be the end of this particular video. Oncogenes. Onco is the prefix for cancer oncologists or cancer doctors, and oncogenes are genes directly associated with cancer. The original ones, as I mentioned, are called tumor suppressor genes. They prevent cancer from starting. You can have problems with the proofreading that you need to have to tell whether a cell is too damaged to divide. You can have failures of a damaged cell. It needs to be able to tell, okay, I'm too damaged to keep doing this, I need to die, apoptosis. And sometimes it's external, where you have your immune system can recognize damaged cells and can send a message into the cell going, you need to die. If those fail, then there's no control that allows these cells to die, or your immune system may actually directly attack these damaged cells. Sometimes a lot of times with oxygen radicals. Remember, we talked about that early on. Something that came out in the last couple of years, something called tissue organization field theory, is connecting cancer to what they call neighborhood interactions, where the cells kind of keep each other, they interact with one another chemically to keep each other normal, and it's a failure of that can produce a cancer. And that, they suggest that may be why, if a cancer manages to metastasize, then it almost becomes exponential because the tumors wind up in places where there aren't those neighborhood interactions and go kind of nuts. It's an interesting idea. They've got a decent amount of supporting evidence for it. The problem you run into with that is that the people that have generated this idea are kind of the opinion that all the other initiations of cancer just aren't worth worrying about, almost like they don't exist. Our theory is the real theory and everybody else is, not. It's gonna make it hard to actually get them taken seriously by other people in the area, right? That's how science partly works. Is it's a community. There's some indication that the interaction between your native bacteria, your microbiome, and your immune system can be critical in controlling and preventing the development of cancer. They're just starting to look at that. And Inflammation is that an immune response in an area where a whole bunch of immune cells kind of descend on an area, the blood vessels expand, area gets red and they leak and it gets swollen. But inflammation itself can be damaging to the local neighborhood. And there's been a lot of attempt to connect inflammation to all sorts of different diseases, diabetes and Alzheimer's and cancer. And so anti-inflammatories might be useful in preventing some types of cancer. There's a lot of analgesics that you can get over the counter that are anti-inflammatory. It may be useful in that. But the research is still very, all over the map. And that's because I'm a big pile of alleles. I'm not the same big pile of alleles as my father was or my son is. We may have a lot of alleles in common, but how many of those associate with cancer? How many of those are important? How many of those are going to react to environmental stresses? Those are all questions that are not easily answered. And so they're starting to look at alleles in different people in terms of how do they interact with chemotherapies, how do they interact with how a cancer will progress. And that study is sort of in its infancy. Again, the people that are doing it seem to be much more certain of what they're finding out than everybody else is. But eventually that avenue will see some pretty decent fruit. Okay, it's a good place to stop.